My name is Nicole Cook, and I'm a master's student in environmental engineering. This presentation will be on Carl Woese, specifically what I found interesting from his paper titled Bacterial Evolution. Here we have the agenda. Carl Woese's background will be first, followed by his life and work, the importance of the work that he did, why people didn't immediately believe what he had discovered, and wrap it up with a conclusion and to cite my sources. Carl Woese was a microbiologist and a biophysicist, and he revolutionized phylogenetic taxonomy. He spent 40 years of his life teaching at the University of Illinois, and his honors and awards include the MacArthur Genius Grant and the National Medal of Science for Biological Sciences Award. So before Carl Woese, we classified life with physical characteristics. Eukaryotes were animals, plants, and fungi. Prokaryotes were bacteria and all of the other microorganisms. So we use the distinction of prokaryotes have no nucleus. To describe something purely in the negative sense is what got Carl thinking that there has to be something more. Through his research, he determined that there was a third domain, and not just two, the prokaryotes and eukaryotes. So in 1977, Carl and George E. Fox discovered the third domain. They defined bacteria, archaea, and eukarya as the third domain of life. With this, there needed to be a new tree of life defined, since there are no prokaryotes and eukaryotes, and prokaryotes didn't just get simply split up. There are three separate domains. So this new archaea. They are single-celled organisms. They have a unique cell membrane. They can also resemble either a eubacteria or eukaryotes, or neither. They were once thought to only occur in extreme environments. So where do they fit in with bacteria and eukaryotes? They were once perceived as strange, atypical bacteria and not a separate domain. However, they have three distinct phenotypes within them. Even though they share characteristics with other domains, they remain very different and distinct. These commonalities prove that all domains shared ancestry, and archaea aren't so strange after all. Here's the original tree of life Carl mapped out to fit the new archaea in. Universal phylogenetic tree determined from RNA sequence comparisons. Line lengths on the tree are proportional to calculated distances. The extent of sequence distance that separates the primary kingdoms is reflected in the degree of phenotypic difference among them. So here's the new tree of life that we use today with the archaea added. Notice the three phenotypes of archaea are displayed. Methanogens, usual coenzymes not in other bacteria, extreme halophiles are the only photosynthetic archaea, and extreme thermophils have at least one unique coenzyme. No immediate known relatives that grow or at or near normal temperatures. So this tree shows all three domains have a common ancestor. Carl Woese stated in 1998 that over time this ancestor refined into a smaller number of increasingly complex cell types with the ancestors of three primary groupings of organisms as a result. Wow, how revolutionary. So how did he prove this? Woese decided to study ribosomal RNAs because RNA is a simpler molecule than DNA, making very difficult experiments somewhat easier. RNA performs the same protein-building task in all organisms. Woese chose to study 16S rRNA, whose function has been constant over great spans of evolutionary time. So after Woese announced his discovery, it was met with very little acceptance. So Woese was seen as an outsider. Textbooks and schools are reluctant to change their ways, and scientists believe that all archaea were just different types of bacteria and not a new separate domain. Several misconceptions held the world back from immediately jumping to accept Woese's work. First is the prokaryote-eukaryote dichotomy. This is the notion that since the 1930s we have had two groups for the living world, either eukaryotes and prokaryotes, and this became the primary accepted theory. Second is the Oerther ocean scenario. This is the theory that the ocean became a soup of energy-rich biochemicals and molecules formed from this. These molecules then somehow became able to absorb organics from the environment, moving through evolution and eventually becoming life forms. This reflects creationism and creates a distinction from the living and the non-living world. The third and final misconception Woese says held us back from initially believing in the new domain was Darwin's little pond. This theory says that relatively simple molecules must have, under certain conditions, been combined and concentrated in a liquid and exposed to some energy source, then producing more complicated prebiotic molecules such as proteins and sugars. So now that these misconceptions are hopefully behind us all, we can move forward to why this is such an important discovery. Overall, this increases our current understanding of evolution and how far back we can look by almost an order of magnitude. With this information, geologists and evolutionists will be forced to have a closer relationship with their overlapping fields. Further, all life on Earth evolved from a single cell or pre-cell. This is in contrast to Darwin and Oerther. Organisms also engaged in horizontal gene transfer instead of competition. So now my next slide is just a huge shout out to Carl Woese for being so persistent in his discovery of life's work. Biology and other disciplines such as medicine will forever be changed by what he found. Here, displayed on the screen, are just a few quotes about him that I think speak to who he was and what he stood for in his field. Wrapping it all up now, we have my sources here, Carl Woese's paper titled Bacterial Evolution and an article from famousscientist.org on Carl Woese. Thank you for listening.